So thank you so much, and I'm really pleased to welcome Fabien Chauveau. Thank you so much for accepting to start this first series of virtual pubs. So Fabien Chauveau has initial formation in the chemical engineering, and he started a PhD in the field of in vivo imaging with a particular focus on neurodegeneration and neuroinflammation, using positron emission tomography, PET imaging. He followed then with a postdoc where he also started to explore the potentialities of magnetic resonance imaging in neuroscience. And now he is a researcher at the Neuroscience Research Centre in Lyon since 2013, and he's exploiting PET, MRI, but more recently he's exploiting the X-ray virtual histology technology to study pathological protein aggregates involved in different neurodegenerative diseases. He has a particular focus also in demyelinating damage to cerebral white matter using animal models for neurological diseases. So please, Fabien, I'm happy to leave you the floor, and we will ask to everyone to switch off their microphone, and if you have any question, you can either put it in the chat or can wait at the end of the presentation.
Thank you very much, Linda and Erika and the whole team for the invitation and the introduction. And I'd like to congratulate the Euro-BioImaging team for organizing this series. It's really a great hub and a great place, and I see a lot of exciting presentations already on the YouTube channel. So today I'm going to try to present you an X-ray techniques which enables to do phase contrast imaging, and I will try to convince you that with that technique we can do a kind of virtual histology. I have one slide on about me and my colleagues. I work in Lyon in France in a big neuroscience institute. And my team is actually located at the CERMEP. CERMEP is an imaging platform, an in vivo imaging platform with MRI and PET machines, which is actually my background. And more recently, I became interested in this ex vivo technique provided by the X-ray and the synchrotron. Most of the work that I'm going to present today is actually Mathieu Chauveau PhD's work. So he should be the one to be thanked for most of the presentation. I will also give some clues about ongoing work by Joshua Gobet, who is also a PhD student, and this is really a collective work and presentation, because I have a lot of material and slides coming from my long-lasting and invaluable colleagues, Emmanuel Brun, who works in Grenoble, and Marlène Viard, who works in Lyon in a different lab. So the first thing is to define or to name what we will be talking about. So we're talking about a set of techniques that are X-ray based, that are using X-ray to produce a phase contrast imaging. And actually, this is not so simple, because if you look in the literature, this will yield a lot of different acronyms. So there is not a single acronym in the literature. And for example, some call those techniques PCCT or XPCT or PCI or hierarchical phase contrast CT. So which is a bit problematic when you are new to this, and there's no simple way to search for the technique on PubMed, for example. So for this presentation, I'm going to use the term, the acronym XPCT. One reason for that is that it was recently chosen by the community as the suffix to describe this type of data as a brain imaging data structure specification. So BIDS is a specification. It's not an image format. It's a specification that you should follow when you want to organize your data so as to share it and to use the same metadata and organization. So I will be talking about X-ray phase contrast tomography, XPCT. What is it about? So I'm going to use my point of view of neuroscientist. Which is, I'm not a developer of this technique, but I'm really a user. So as a neuroscientist, I am used to perform histology, which consists in cutting the brain and doing microscopic sections, and to use staining or antibodies to reveal a particular contrast. And in this case, we will be able to scan the entire piece of samples. So here, an entire brain of mice, and we will be able to navigate into the brain, so to virtually slice it. And you see that even if this brain is unlabeled, so it's only a tissue that is chemically fixed, there is no labeling, you see that there is already a contrast. So we will be able to like digitalize the biological sample, and then to perform virtual slicing and virtual histology. This is possible because with the X-rays, we are not measuring the absorptions like you do when you go to the hospital for a radiography or a CT or a scanner, or when you use a CT scanner in the lab, because you can only measure the absorption. In this case, we are going to be sensitive to the refraction, and refraction is actually a small deviation of the X-ray light when you come from one medium to another. So this is what you experience visually when you look at this glass. You see here a break that is a kind of illusion because you are moving from water to air. And physically, this is going to translate into a shift in the phase, whereas in the case of standard radiography, you are sensitive to an attenuation of the signal. And of course, the beauty of that is that you are going to be sensitive to the microstructure even in soft tissue. In a soft tissue like the brain, there is almost no absorption contrast because it's a soft tissue. But with the phase contrast, we are going to reveal the microstructure, and we are going to be sensitive to microscopic interfaces in the microstructure. How to do this? Actually, there is quite a simple way and a simple device to do this. To explain this, you have to think that if the detector, the screen, is close to the sample, then the contrast will be dominated by the absorption. But if you introduce a propagation distance, then you will allow the refraction to produce an effect, and this effect will be measurable in the terms of interference fringes at the edge of the object. So this is the most simple, this is not the only one, but this is the most simple way of being sensitive to phase contrast and not only to attenuation. But to do this, you have to have an X-ray source with a very high spatial and temporal coherence, which means that you cannot do this with an hospital or a laboratory source, but you have to go to a special infrastructure, which is a synchrotron. And with this kind of physical properties, so spatial and temporal coherence, you are going to be sensitive to fringes. This is really what you see when you look at the bottom of the swimming pool on a sunny day. You have a distortion of the light when you go from the air to the water. Just a few words on a synchrotron. Synchrotron is an electron accelerator. So you have actually electrons that are traveling here in a storage ring, and because their trajectory is not linear, you will have light emitted tangentially to the ring. Though this is called a beamline, and each beamline is characterized by a specific light that is emitted. Some are using X-ray, others are using UV or infrared. And in each beamline, you will have different etches, experimental etches, and a control room, of course. Here is a map of the two synchrotrons that are located in France. One term, Soleil, is located near Paris in Saclay, and the other one is the European Synchrotron Radiation Facility in Grenoble. So you see that the number of available beamlines at each facility is quite high. Of course, not all of them allow to do X-ray phase contrast imaging. Not all of them use actually X-ray. I can mention the anatomics beamline at Soleil, where you can do phase contrast imaging, and the BM5 beamline, for example, in Grenoble. This is a view of the inside of a synchrotron, and you see here the beamlines that are the arms around the storage ring. And those are some pictures taken on the anatomics beamline and on the at the ESRF on another beamline, which is now closed, unfortunately. So roughly to show you that, of course, the environment is a bit striking and surprising for a biologist, but this is the place where you have your sample, and this is the detector, so the camera, and this is the way where the X-ray are going to travel here and here. So a very complex environment for a biologist, but actually the experiment from the use point of view is quite simple. You have to go there with your collection of already prepared samples. So this is one of my collection of mouse brain. Those brains were chemically fixed, though animals had a intracardiac perfusion with formaldehyde. And we also use, and I'm going to talk about this later, we usually dehydrate them in ethanol. And then the acquisition itself consists in rotating the sample. Is going to make a full run, and during that you will acquire a lot of projections. A lot mean several thousands of projections. So as a user, what you have to do is position your sample on the stage, and then go back to the control room and align, which takes only a few minutes, and then launch the scan. And a scan is achieved in less than 5 minutes. We are talking about a pixel size that is below 10 microns and could even reach 1 micron, with a field of view that is typically around 1 centimeter. This is the kind of energy and distance between the sample and the detector that we use. So this is actually very fast. You need a bit more time to reconstruct the 3D volume, which usually takes 10 to 50 minutes. Then one challenge is that you end up with really big data. So one row reconstructions will be typically 100 of gigabyte. And when you optimize your field of view or crop your image or compress it a bit, you usually end up, I'm talking of a mouse brain, you usually end up with an image that is from 10 to 20 gigabyte. So it's quite big. And if you really regularly go to the synchrotron, you end up with that picture, which is a real picture from one colleague, and you have quite a huge work or a huge amount of time to spend to sort the data, to organize, to correctly tag them, and try to compress. I'm insisting on this because it seems obvious, but you actually spend a lot of time to do, and maybe you're spending a lot of time at this stage than on the acquisition itself. So this is the type of image you end up, and I will show you two different structures that we chose to study. The first part will be dedicated to myelin, the study of the white matter in the brain. And the second part will be dedicated to the amyloid beta plaques that you have in Alzheimer's disease. And most of the data that I'm going to show is from a pair of papers that was published 3 years ago in Biomedical Optical Express. A few words on the initial experience. So I said at the beginning that I actually come from the in vivo imaging field, and our first experiments at the synchrotron was actually with the goal of detecting MRI contrast agent. I'm talking about iron oxide particles that you use for MRI. Those are MRI imaging. And we wanted to have a closer look at the fate of those nanoparticles. And because they are metallic iron, they were also detectable with X-rays. And then that was our first good reason for going to the synchrotron and to perform this high-resolution ex vivo imaging. Then we realized that we could manipulate the contrast between gray matter and white matter just by dehydrating the sample into ethanol. And if you go to 50 or 100% ethanol, you get a really nice contrast, and the white matter is now white in hyper signal. So what happens is that you make the overall tissue more dense, and you change the refraction indices, and you optimize the contrast between white and gray matter. So obviously, okay, first this is a video of a typical scan. After cropping, you see the tube and reorienting the images so that you have the usual coronal slices. You see, of course, the great white matter contrast. You see the little dots in the hippocampus that are the amyloid plaques. So the first idea was really to use this fantastic contrast, showing you the white matter in 3D with a very high resolution, because the pixel is isotrope. So you get a pixel size of typically 5 or 7 micron in every direction. This is in contrast with a technique like MRI, for example, where you typically have a good resolution in plane, but a very thick slices. A few cases where you can use this, a few medical cases where you can use this white matter contrast. Here in a stroke model, so the lesion itself, which was necrotic, went away when getting the brain out and after the perfusion, but The necrotic part is not the interesting part. You can see here that the adjacent white matter has a loss of contrast, which indicates that they have a white matter damage adjacent to the stroke lesion. Another model from colleagues in Lyon was a postnatal hypoxia model, which results, and it's quite well known, in a defect in myelination, and we were able to quantify this defect of myelination using this imaging technique. And there are two arguments to say that this white matter contrast is actually caused by myelin. The first one is that if you use a brain clearing technique, you lose completely this contrast, and this is because brain clearing usually proceeds by removing the lipids, so removing the myelin sheets. And the second one is from this beautiful study where they actually scan the same brain at different stages during paraffin embedding. And you see that the white matter contrast is still there after the ethanol step, but then disappears after the xylene step, which is not to remove the lipids, and there is no more white matter contrast when you look directly into the paraffin block. So those are good reasons to think that we are actually looking at the myelin, and this is the change in refraction index between the gray matter and the lipid-rich white matter, which creates this nice contrast. Then we worked with a company in Grenoble called Novitome to try to derive or to recover the orientation of the fibers, just like you are doing this with diffusion tensor imaging. So here you have a comparison that is not totally fair, because this is ex vivo imaging at the synchrotron, and this is an in vivo image from the MRI. So obviously the resolution cannot be compared, but I want here to make the point that we are able to get the same outputs, which are the fibers direction, and they are here color coded. And from diffusion tensor imaging, you can extract quantitative parameters such as fractional anisotropy and axial and radial diffusivity. And we were able to compute the same DTI-likes parameters. So of course, it's not through fractional anisotropy and diffusivity, but it's the equivalent that were derived from the XPCT scan. And I would like to insist again on the fact that we have a very good resolution in the three directions. So not only in the coronal plane, but also in the axial view of the brain. And the last thing, which is really work in progress, it's quite obvious when you have the orientation, then you want to do what people do with DTI. You want to do a tractography of the white matter tracts, and this is a first version of the tool, a first preliminary result that we published this year as an conference proceeding, and this is the work of Joshua Gobet, and he is now able to produce tracks with the correct orientation. You see that they are in this case really small broken tracks, but now he has better results concerning the length of the tracks. But already you can see that at this level, in the region of the fornix, they are crossing fibers that usually you cannot resolve with MRI because you don't have enough spatial resolution. And here it seems that we are able to resolve the different directions of those crossing fibers. So this is really an ongoing effort from my group, but not only other groups are working on that to really being able to make a high resolution tractography from those brain imaging. Now I'm moving to the second part. I'm going to talk about amyloid plaques, which is one of the main hallmark of the Alzheimer's disease. And I want to remind you that how pathologists proceed when they have to make a final diagnosis, a postmortem diagnosis of Alzheimer's disease. They have to sample the brain and they have to go for paraffin embedding, then they have to slice, then they have to stain, and then they look at the sections and they score. They give a score to the amyloid pathology, given the morphology of the plaques, the number, and the number of areas that are concerned in the brain. So it's quite a fastidious and very long work just to ascertain the diagnosis of Alzheimer's disease, or at least to ascertain the fact that there was an amyloid pathology. And so the goal here was to explore this image modality to make this process or to show the proof of concept that we can actually detect those amyloid plaques with explicitity, and maybe define more quantitative parameters to characterize this pathology. There were previous studies on amyloids and even like 15 years ago. So this is a good opportunity for me to say that the technique in itself is not really new, even if at that time the way to acquire the phase contrast imaging was not exactly the one that I described, but we were already able to get those type of images, and people already noticed that amyloid plaques could produce contrast. Then more recent work by the group of Paula Cohen, who used different beamlines with different spatial resolution to get down to the nanoscale to look at those amyloid pathology in 3D, and great work from the Team Saldik group in Germany that look at human tissue. First look at human tissue and identify plaques-like structures in the human brain. But those studies were still quite qualitative, and we wanted to bring more quantification to the data. So we started by gathering a few brains from different amyloid strains. If you work in that field, you know that there are a lot of transgenic mouse models that have been generated with mutations that have been identified in familial cases of Alzheimer's disease. So this one has mutation on the APP gene, this one on APP and PS1, and this one is a triple transgenic mice with mutation on the tau protein as well. We looked at the hippocampus because we know this is where the pathology starts, and even visually you can really notice that amyloid plaques first are detectable with X-ray phase contrast tomography and have a very different appearance. In this one, you have small and bright dots. In this one, you get a more diverse shape, but you can recognize the usual shape, I would say, in humans, where you have a core and a corona around. And in the triple transgenic brains, we had less deposits, but they were much bigger and much brighter than the other ones. So to put numbers on that, we had first to develop a 3D segmentation pipeline, and Mathieu Chauveau, during his PhD, succeeded in doing it with Fiji, which is a popular tool for a biologist. He used a combination of plugins the segmentation editor, the Weka plugin to do machine learning, and so to learn so that you learn the computer how to properly segment your plex, and then other plugins such as the MorphoLibG from David Legland, which enable you to extract a set of 3D parameters. So this pipeline is really described in detail and available on Zenodo, and you could actually use it for any kind of 3D segmentation. What is important here is that you end up with a new set of parameters. So I don't want you to look into details to the results. There were obviously differences between the strain, but the thing is that the parameters are not the usual ones. We are talking about sphericity or flatness, and of course the volume. So we are talking about 3D parameters that characterize the shape of the aggregate, and those parameters are not available when you are doing standard 2D immunohistochemistry. So we are describing a new set of parameters which with which you can really quantify the shape of your amyloid plate. Another way to look at those results is to look at the correlations between those different parameters. Of course, they are not independent from one to another, so they are correlated, and it's interesting to note that from one strain of mice to another, the correlations are actually different. And a final way to look at the same results is to get back to the 3D images, and you see the hippocampus that was segmented in a semi-automatic way, and then you go back with your segmentations, and here the plaques are color-coded with one of the 3D parameters, one of the shape parameters, which is the sphericity in this case. So I'm going to launch back the presentation and back to the place. Sorry about that. And then the question we had is obviously we wanted to understand why the amyloid plaques were actually detectable with the XPCT and why they actually produce contrast. And so for that, we had two hypotheses. We know that those plaques are made of insoluble fibrils, so that is maybe a reason to change the refraction indices and to produce a contrast. We know also that those amyloid plaques tend to entrap biometal, endogenous metals such as copper, iron, or zinc, which is also another reason for producing a contrast. So to try to understand and decipher this contrast, we designed a study that was entirely done at Soleil, at the synchrotron, but for which we had to use 3 different beamlines. So we had to get some samples, animal samples, and also human samples. We had to separate the hemisphere. One hemisphere was dedicated to imaging, so you only need a chemical fixation and dehydration to do that. And the other hemisphere was sliced and then put on membranes so as to be used on another beamline using infrared spectroscopy, and the same sections were then used on a third beamline to perform X-ray fluorescence. So the infrared spectroscopy will give you, when you look at the AMID1 peak, will give you a ratio between beta sheet structures and alpha helix structure, not alpha sheet, but alpha helix. And of course, those amyloid fibrils are enriched in beta sheet structure. And the X-ray fluorescence enables you to map with a very high spatial resolution different type of metals, such as the copper, zinc, and iron. I show you first the results in humans. So we had samples of cortex from one case of Alzheimer's disease. And you see that actually the amyloid plaques are not detectable on those images. What you see here, the small dots, are the neuronal bodies, and the blue arrows here stand for amyloid corpusculaceae. So it's not amyloid plaques by themselves, but structures that you typically find in old people, but are not a non-specific marker for aging. And of course, we couldn't really see the expected amyloid contrast that you get with standard immunohistochemistry. But we had also an inherited case of Alzheimer's disease, so a familial case of Alzheimer's disease. And in this case, we could detect some amyloid deposits, which are those large and bright spots among the cortex. And then when we look at the other techniques, so the infrared spectroscopy showed us that the genetic case has a much higher level of beta sheet than the sporadic case. And also this genetic case had a much higher level of metals that were entrapped in the plaques compared to the sporadic case. So with this human data only, we couldn't really understand what was the reason for the detection of amyloid plaques only in the inherited case and not in the sporadic case of AD. So we had to went back to animals. We had another collection of mouse strains, not exactly the same strains as the study before. And it was really interesting to observe that we actually get contrast that we already observed, those bright spots here in this mouse strain, but also new contrast. We had in those two cases amyloid plaques that had a dark rim and a bright spot in the center. And we also had some samples of a transgenic rat, and in this case they had only bright spot, just like the first mouse sample. And so when going back to the 2D acquisition and the infrared spectroscopy, well, on that part, what we observed is that in all animal models, there was quite a similar level of fibrils, which are beta sheet structures. They were all the plaques were enriched in beta sheet structure, which is expected, but they were enriched at a very similar level. So this quantification, this factor could not help us to discriminate between the strains and to explain the different contrasts. And in contrast to that, the X-ray fluorescence spectroscopy showed us that the amount of metal levels that was entrapped in the amyloid plaques was actually highly variable between the different strain of animals, elevated, of course, compared to the wild type animals. And this overall level actually correlated with the imaging findings. So this is quite a complex figure to summarize the finding that in every cases in the animals where you have a bright spot, such as here or here in the middle of the dark rim, what you get is a strong accumulation and a focal accumulation of metals. And actually the size of this accumulation correlated not with the overall size of the plaque, but only with the bright core. And this was actually consistent with our observation in the genetic case, because in the genetic case of AD, we had a bright core and a bright rim around this, and this was consistent with the X-ray findings that you have quite a large region where the overall amount of metals is elevated. So our conclusion on that is that a positive contrast on the XPCT, a bright spot on the XPCT, is mainly explained by the accumulation of metals in the amyloid plaques. So we don't have the full story, and I'm not able today to explain why some animals have a dark rim around the bright spot here, while others have not. And it's still not totally clear why in most sporadic AD patients, we are not able to detect the amyloid plaques. One reason is probably that the plaques are more diffuse than in this obviously very severe genetic case. And I'd like to finish with again with an ongoing work on the amyloid pathology. We are now looking to cerebral angiopathy amyloid, which is a pathology where the accumulation of A-beta peptides occurs directly in the vessels. And looking again at this inherited Alzheimer's disease case, we have vessels here that are really bright, and we see also downstream effects of this angiopathy amyloid pathology, which is the enlargement of perivascular spaces. And in this case, Mathieu was able to properly segment the vessel wall on one side and the perivascular spaces on the other side. So we expect to again derive new 3D biomarkers that are not currently available when you do traditional immunohistochemistry, where you have to cut the vessels and you usually don't control the incidence of the slicing. Just a few more slides to conclude if I have more time, but just to show you that I focused on two scientific cases, myelin, amyloid pathology, but there is a lot of pretty interesting observation that you can make even on the mouse brain. For example, in some animals, we are able to see subregions in the cortex. The contrast in the hippocampus is really intriguing, and there is, I think, a lot of work to do to properly explain why we get this contrast and why we are able to have those nice images of the hippocampus anatomy. Also, the study of the vessels is possible with this technique. You see here extracerebral vessels, such as the circle of Willis, but also intracerebral vessels in this animal where the blood has been removed by the intracardiac perfusion. Also, the cerebellum is will always give you really nice images where you are able to differentiate the different structures. And finally, beautiful images of choroid plexus, which you can actually look at and maybe segment in 3D. And I know there are work ongoing from other teams on that topic. It's not really a perspective, but more recent work or directions that we are now going to. I presented work where we were able to look at quite small objects such as a mouse or a rat brain. But now in some beamlines, you are able to perform this technique on quite a large object, and we've been able here to scan a whole macaque brain in Grenoble quite recently. And there's also a consortium, the Human Organ Atlas hub that is led by Peter Lee at UCL, that scans human organs entirely, so not only brain but other organs, and you can find a lot of great videos from their work on the internet. And on the other side, there are also people working on the ability to perform XPCT not only at the synchrotron but with a conventional sources, which would open up the field by being able to do this with a laboratory scanner. So there are a company doing this, and we have the talk of Etsyscope after my talk. But there is also an academic effort on that. I want to mention the Musitox platform in Grenoble, and there are several ways to do that. I just took this figure, and one way to being able to do that without the coherence of the flux of a synchrotron is to use a membrane and to perform the acquisition with and without the sample, and to see how the membrane image is affected by the refraction from the sample. So I really think that in the near future we will be able to use XPCT, but not only at the synchrotron. And more generally, I think that this is really a great technique, because the kind of resolution that you get, which is typically between 1 and 10 micron, is exactly the gap that you usually have when between in vivo imaging techniques like MRI and PET and a wealth of microscopic imaging technique, optical techniques, where you usually work at a sub-micrometric resolution. So I think there is a great potential for multi-scale imaging, or what people now call transpathology in the future. So this is my final slide. For the moment, to perform XPCT, you have to go to the synchrotron. And from what I show you, you may have the idea that it's really complicated to go there. It's actually not. Every synchrotron has a call, which is launched every 6 months. And what you have to do is write a 2-page proposal. Of course, for that it's good to first get contact with beamline managers so that they guide you. But if your project is selected, then the access and the beam time is free. So it's not that difficult, and it's actually not an expensive technique. One hub I'd like to mention for meeting the community is the annual meeting of the European Society for Molecular Imaging, where we have an X-ray study group and a lot of people using standard X-ray, but also doing phase contrast imaging. And to conclude, we would really like to that more biologists or neuroscientific follow us on this road. At the moment, this XPCT road is a bit dark because there's a lot of things that we don't understand about the contrast and the analysis of those big data is also a bit tricky, but there are a few lights now, and we really would like you to help us to enlighten the way and make the full potential of this great technique. I thank you for your attention, and I'd be happy to answer your questions.